Hello students, welcome to EPG Patashala. I am Gaurinath from School of Life Sciences, Jawaharlal Nehru University. Today we are going to discuss about a module on structure and function of actin and myosin. In this module, we will learn about cytoskeleton and we will learn about the structure of actin, its dynamics and structural changes in actin while going from G-actin to F-actin. We will also learn about myosin structure, Powerstroke, and the structural changes in myosin in the process of Powerstroke. Cytoskeleton. As I showed you in earlier image, the network of tubulin, actin filaments and intermediate filaments are called as cytoskeleton. These three filaments are protein structures which give a shape to the cell, and that's why they are called as a skeleton of cytosol or cytoskeleton. Except intermediate filaments are little more, they have longer life structurally. The actin filaments and tubulin filaments are more dynamic. The function of cell depends on the dynamic network of these proteins. So the dynamic network of this protein will influence the functions like crawling of the cells, muscle contraction, changes in the embryo, developing embryo, transport of the organelles in the cell, segregation of chromosomes and endocytosis, etc. These activities basically depend on actin filaments, microtubules and intermediate filaments. As I said, intermediate filaments have little longer life. At the only majority cases, only at the time of cell division they completely dissolve and new intermediate filaments are formed in the baby cells. The actin filaments and microtubules are changed drastically depending on the function of the cell and activities of the cell. There are several accessory proteins and motor proteins which function, which crawl on these filaments. Like example, an actin filaments, the myosins walk. Kinesin walks on tubulins. So tubulins becomes like railway tracks and kinesin is like a train which goes on these tracks. So all these proteins, a network of proteins which influences the function of the cell. Actin. Actin is found in all eukaryotic cells and it is one of the most abundant protein in all cells. In cells, total protein component of the actin varies from 5 to 10%. In muscle cells, actin comprises up to 10% of the total weight of the cell protein. In non-muscle cells, actin makes up to 1 to 5% of the total cellular protein. Now, the actin component varies from the cell to cell and the function of the cell which it requires. Actin can exist as a globular monomer called G-actin and filamentous polymer called F-actin. Actin polymerization. Polymerization of G-actin has three sequential steps. So the G-actin polymerizes. First, it forms nucleation. Nucleation marked as a log period in which 2 to 3 G-actin molecules act as a seed or nucleus. So that is basically a small nucleus of F-actin is beginned. The elongation phase in which the seed rapidly increases in the filament length by the addition of G-actin to both of its ends. So once the seed is formed, once the nucleation is formed, the actins attach to the nucleus and elongation takes much faster. The third is the equilibration phase or steady state, in which growth of F-actin is precisely balances the dissociation of the monomer from the G-actin. That means the number of actin monomers attached to the F-actin and the number of G-actin dissociated from the F-actin remains equivalent. So that is where the steady state of the length of the filament is maintained. Here I have shown the model how the actin filament assembly takes place. So the G-actin, that's actin monomers, have shown as a single uh, round shaped structures, slowly forms dimers and trimers. And in this state, the formation of dimers and trimers is much slower, and this is called a nucleation. And these nucleus, once formed, then the actin filament elongates. Now you can see the F actin at the end, where the barbed end, from where the G actin associates much faster, and the pointed end where the G actin actually dissociates. But in the beginning, the association could be from the both sides. But once the F actin reaches the stationary phase, the actin adds at the barbed end and dissociates at the pointed end. At steady state or stationary state, the actin filament where the actin is attached and detached is also called as thread milling. At barbed end, actin is attached when it is bound to ATP. So ATP actin is added at the barbed end, and as the time increases, as the length of the polymer increases, somewhere in the middle the ATP is dissociated. Then the actin stays as ADP PI actin. But the transition state of ADP PI will not stay for longer, and the PI is dissociated from the actin. At the other end, the ADP actin stays, and from the minus end, or it's also pointed end, slowly the ADP actin dissociates. After this dissociation, the ADP is exchanged with ATP, and the ATP actin again is ready to go and bind at the barbed end. And this cycle is actually what we call as a stationary state or thread milling. This actin dynamics, the thread milling, is not simply done by itself. In the cell, there are several proteins which help this thread milling process. In this slide, I have shown you many proteins which participate in this actin dynamics. The ARP2-3 complex, or it's also called ARP-warp complex, is actually generally helps in the nucleation. And there are several capping proteins which stop the growth of the actin filaments. And there are many proteins which also stabilize the actin filaments. Then you have several ADF covalent proteins which are called actin depolymerizing factor proteins, which help the actin to depolymerize and exchange the nucleotide. Then we have a profilin which takes the actin to the barbed end and associates to the F actin and helps it to grow. So there are several proteins which helps in this actin dynamics and thread milling. So the growth of actin filament at one side is basically as a directional where a cell wants it to grow, and also at the other end where it dissociates on the basis of the cell functional requirement, it dissociates. You can see in amoeba the pseudophore formation and amoeba walking is regulated in one direction. So the direction where amoeba is moving, the actin filament is growing, and the other side where amoeba is retracting is actually where your actin filaments are depolymerized. Here I have shown you the G-actin structure. The G-actin structure, it can be divided into 4 subdomains. The subdomain 2 is the smallest one, and subdomain 1 and 3 are very similar, which indicates a gene duplication event. Subdomain 2 and 4 seems to be a large insertions in the between 1 and 3, causing this structure. I also showed you the nucleotide binding cleft between domain 2 and 4, and there is another cleft between the domain 1 and 3. The cleft between 1 and 3 is generally where other proteins bind for the regulation of actin depolymerization and polymerization. So, the many proteins which bind between domain 1 and 3. The domain 2, there is a large loop which is called as the DNase binding loop. The DNase which binds to this loop stabilizes G-actin as a monomer after binding to DNase. Now, in the F-actin structure, this domain, the domain 2, is expected to have a large structural change. Also, there are other small changes in other domains, but the domain 2 is expected to have large structural change. Structure of D-actin. As I described earlier slide, G-actin has 4 domains, 1, 2, 3, 4. The 1 and 3 are larger domains and 2 and 4 are smaller domains. All these 4 domains are stabilized by hydrogen bonds, salt bridges, and phosphate groups of ATP. They do interact, all the 4 domains do interact with ATP, and ATP sits in the center of the 4 domains. ATP and calcium are centrally located in the actin monomer. Towards the subdomain 1 and 3 is called barbed end, and towards the subdomain 2 and 4 is called pointed end. Most of the G-actin binding proteins bind to the barbed end between the domain 1 and 3. So there is a huge groove where generally a helix enters into these groove between domain 1 and 2, 1 and 3 of the actin. The large loop in the subdomain 2 interacts with the DNase 1. This large loop of subdomain 2 changes its structure when converted from G-actin to F-actin. The structure of F-actin. Here, the structure of F-actin model is shown here. The filamentous actin, which we call as F-actin, addition of cations, magnesium or calcium, induces G-actin to form F-actin much rapidly. The polymerization of actin or formation of F-actin classified into 3 steps as we discussed before: the nucleation, elongation, and equilibrium. Here you can see the F-actin structure where one monomer sits over the other monomer. So it's like a 3 come together to form a nucleus, or you can also take 2 as a nucleus. Basically, it's a 2 parallel kind of actin polymers which twist itself to form a longer F-actin structure. It will be good if you can see the PDB 3LUE in 3-dimensional structure. That will give you a much better clear picture of how actins are polymerized to form F-actin. Here I have shown you 2 actin structures, one with ATP, the other with the ADP. So the ATP state is expected to be the G-actin state, and ADP state which is expected to be similar to F-actin state. And you can see there is a clear difference between the subdomain 2. The subdomain 2 in ATP state is actually loop, and the ATP state is also was determined in complex with DNase 1 loop. That could be the other reason why you have a domain 2 is a large loop. In the case of ADP state, you can see in the domain 2 is actually helix. So the loop which actually converts into the helix, and the domain moves closer to the domain 4. Now we move to other protein of the muscle or cytoskeleton is myosin. Now the myosin, the easiest example is you can see from the muscle structure. If you take out a single fiber, muscle fiber, and look into the electron microscopy, you can see the light bands and the dark bands. The light bands are made up of actin filaments, and the dark bands are made up of myosin filaments. The dark myosin filaments, which forms bundles, and they slide and the actin filaments. And when they slide, and that is basically responsible for muscle contraction. The sliding of actin and myosin over each other is due to contractile cycle. Myosin, which is bound to actin when the new nucleotide is bound to it, that's called rigor state. When ATP is bound, the myosin detaches from the actin. When the ATP is hydrolyzed, there is a structural change in the myosin, and that can now bind to actin filaments. As soon as it binds to actin, interacts with the actin, it induces a structural change in the ADP phosphate myosin, and that can opens up the pocket where the phosphate can be released. And this causes a power stroke where the myosin structural change takes place. Now that causes the movement of actin compared to a myosin, and this is where actually the sliding of filament takes place. Then ADP is released where the myosin now binds strongly to the F-actin and comes back to the rigor state. Now this cycle continues and causes the myosin movement over the actin or the muscle contraction. To study the structure of myosin motor, there are several ways of preparing the myosin motor. In conventional way, the myosin filaments are taken and proteolized with different proteases to prepare subfragment 1 or motor domain. The large myosin motor filaments are called two-headed myosin and that are digested with trypsin, which leads to a soluble heavy meromyosin. These heavy meromyosins have two heads, the 4 light chains and a long filament. For the structural studies or for the crystallographic studies, further digestion is done with a papain, a regulated digestion, which leads to subfragment 1. Now, subfragment 1 has a whole motor domain and the 2 light chains, and the long coil domain, S2 fragment, is separated out. It could be further digested by proteases, which gives only regulated domain and the motor domain separately. Now, most of the studies in earlier times, the structural studies are done on subfragment 1. In the last few years, as the molecular biology techniques improved, the myosin could be cloned and expressed in the insect cell or dictyostelium. But earlier, all the myosin structures were done from the natural sources. Here I am showing you the myosin structure in no nucleotide state. The no nucleotide state is expected to be a rigor-like state. But the structures which told the opening of the actin binding site, so therefore it is called a near rigor state. The first structure to be reported by Raymond et al. In 1993 from the straighted chicken myosin S1. And later, the near-rigor conformation structure is reported from the scalloped S1. The scalloped S1 was considered more functional compared to the chicken straighted S1. You can clearly see the myosin head could be divided into 4 subdomains. The upper 50 kilodalton domain, lower 50 kilodalton domain, N-terminal domain, and converter. And there is a long helix region which is bound to 2 light chains, that's called a regulatory domain. These 4 domains are connected by a small loops and a relay. Now these loops could be called as switch 1, switch 2, SH1 helix and relay. The structural changes due to the nucleotide hydrolysis and binding could be structural changes in these loops. We will discuss more as we see the other structures. Myosin structure in pre-Powerstroke conformation. This is a conformation when ATP is hydrolyzed but the phosphate is still there in the pocket. That means both ADP and phosphate are still in the active site and the phosphate is not yet released, and that is called a pre-Powerstroke conformation or an intermediate state. Now here, earlier structure which is reported from the smooth myosin state 2 with aluminum fluoride, ADP aluminum fluoride, and later from the scallop S1 with ADP vanadate. The scallop one had a 2 light chains, but the smooth muscle myosin had only 1 light chain. But both the structures give very clear changes as we have seen from the contractile cycle. Now you can see there is a 50 kilodalton lower subdomain which is rotated to move the converter and causes the change in the conformation. I will show you all the 3 different conformations where the structures are determined, and we will discuss about the Powerstroke again. The ATP bound state are where the SH1 helix is melted. The connection between the motor domain to the liver arm is only seen in the scallop S1. Now here there are several discussions about the probability of this structure state in the contractile cycle. If you look at the structure, here the upper kilodalton domain, lower kilodalton domain, N-terminal domain are exactly same place, but the SH1 helix is melted and the connection between the motor domain and the liver arm is lost. Now this structure is represent the ATP bound state where the structure is wobbly and it should not transmit when the nucleotide is bound the unproductive movement of myosin in contractile cycle. Here I am showing you the structure of scallop where it looks like actin bound state. When the nucleotide is removed, which was earlier determined as a near rigor state, in this structure the cleft between the upper 50 kilodalton domain and lower 50 kilodalton domain is closed, and the nucleotide binding path, the front door that between the N-terminal domain and upper kilodalton domain is more open. So this clearly shows the actin bound state or rigor state. Now here, once the lower 50 kilodalton domain and upper kilodalton domain come closer and bind to actin, the nucleotide binding pocket, which is also called as a front door, now is more accessible to exchange the nucleotide from the ADP to ATP. Here, the rest of the structure is very similar to near rigor state, but only the difference is the movement of the upper and lower 50 kilodalton domain and in relation to the N-terminal domain. Here I have shown you only the domain structure and all the 3 conformations at the same place. Mind you, the near rigor state and rigor-like state are very similar in terms of domains except the upper 50 lower 50 compared to the N-terminal domain. To understand the Powerstroke, you need to understand these 3 different states. In the nucleotide free state, you have upper 50, lower 50, N-terminal and converter in a most relaxed state where the nucleotide could be exchanged. Once the ATP is bound, you see internally uncoupled state. The 50 kilodalton lower domain which moves away from the N-terminal domain and causes there is a distance between the 2 domains which causes the SH1 helix contacts are lost and due to which it is melted. The SH1 helix generally have 2 glycines in almost all myosins, which makes it very flexible helix and which melts. And here, because of this, we expect the unproductive movement of myosin is not caused. As soon as the ATP is hydrolyzed to ADP and phosphate, it changes the conformation to pre-Powerstroke state. Now here the nucleotide binding pocket is closed. The phosphate cannot move out of the active site cleft, and therefore there is a structural change where the phosphate interacts with switch 2. I have further slides which will show you how the conformation is caused. The lower 50 kilodalton domain, which rotates as indicated by arrows, which causes the converter to move, and this causes the lever arm to change its structure. The scalloped myosin S1 is the one only myosin which is determined in all three conformations. Here we have superimposed the motor domain. The small structural changes in the motor domain are ignored, and we have only shown the orientation of the converter related to the motor domain. You could clearly see from the internally uncoupled conformation to Powerstroke there is a huge difference. It's more than 90 degrees difference, and which causes this movement. So the small nucleotide which is bound and hydrolysis is taken place in the active site is transduced to the lever arm, which causes a large structural change. In this slide we can clearly understand the motor mechanics. Here the most of the structure is removed and only the active site components are kept. The nucleotide is bound between the P loop, switch 1 and switch 2. The ATP bound state or ADP state cannot interact with the switch 2, which is far apart. But once the ATP is hydrolyzed, the ADP PI is formed, the phosphate which has to move from ADP and which interacts with the switch 2, which causes the switch to move closer. And these small changes which converts the converter movement and large structural changes in the lever arm, which we have seen in the previous slide. Here from the nucleotide binding site, and if you go further to the power transmission site, are joint movements. Now you can clearly see three conformational states where the relay, SH1 helix and converter orientations. So the relay which actually pushes the converter to cause the structural changes in the lever arm, and you can see that switch 2 which is connected to lower subdomain through relay, and that causes the structural changes. On the basis of these structures we have shown you here and several structures which are reported by other groups, several motor mechanisms are being proposed. I find this mechanism is more easy for the students. Here the myosin motor is represented as a simple structural transmission. You can see the SH1 helix which has two glycines and which is termed as a clutch here. So once you press the clutch, you don't change the structure, the gear movement into the productive movement. So when you have ATP bond, the SH1 helix which melts, and the ATPase, the nucleotide binds and releases, you can see at the center. The upper 50 kilodalton domain and the lower 50 kilodalton domain is closed and open as nucleotide is bound or it's bound to actin. You can also see the arrow marks indicating the lower subdomain and the relay movement which causes the converter to move. So in short, this motor components which explains the motor mechanism of the myosin, which gives the myosin movement and the actin filaments. Now after the informations which we got from the structures, we can go back and look at the contractile cycle again. So when there is no nucleotide, the myosin is bound to actin very strongly. This is called rigor state, where if you remember, the upper 50 and the lower 50 kilodalton domain come close and the nucleotide binding site is open, and that's how the nucleotide exchange takes place easily. and where the ATP enters into the myosin. Now once ATP enters into the myosin detaches from the actin because the upper 50 kilodalton domain, which moves closer to the N-terminal domain, and the upper 50 lower 50 cleft opens up. This causes dissociation of myosin from the actin. Now here, which actually it moves the myosin into internally uncoupled state. Once the ATP is hydrolyzed, it moves to pre-Powerstroke conformation. The pre-Powerstroke conformation is now can bind to actin. Now it slowly binds to actin, which causes the phosphate release and the Powerstroke. The association of actin with the myosin in pre-Powerstroke conformation, which accelerates the Powerstroke or hydrolysis of ATP, release of phosphate. Now this causes the movement or the sliding of myosin and the actin filaments. Once it binds strongly, the ADP is dissociated and it reaches the strong actin bound state. Now this contractile cycle, once you have all the structures which could understand the motor mechanism and the structural changes in the myosin head. Okay, now I summarize this module of structure of actin and myosin. Cytoskeleton, which gives a structure to the cell and plays a major role in the shape and function of the cell. As I said before, the skeleton which gives the shape to cytosol is called cytoskeleton. And actin forms a dynamic network of filament structure, is one of the major component of the cytoskeleton. The tubulin and intermediate filaments are other components of cytoskeleton. Actin has 4 domains and ATP binds in the center. There is a major structural changes in domain 2 and domain 4 a little bit when it is changing from monomeric state to filament state, that is G state to F state. We call G state because it is a globular state and F state is a filamentous state. There are several actin binding proteins participate in actin dynamics, that is moving from G state to F state. Most of the proteins, they bind between the domain 1 and 3. There is a groove which has a hydrophobic, largely hydrophobic surface, and the actin binding proteins majorly with the helix enter into the groove between the domain 1 and 3. There are few proteins which bind to domain 2. In domain 2 there is a large loop which changes its conformation from loop kind of state to helix kind of state when it interchanges from the globular state to filamentous state. This helix actually enters into the groove between 1 and 3 of another actin just above that, and that's how the F actin is formed. This F actin could be disassembled or depolymerized with several accessory proteins which bind to G actin, and this dynamic process is controlled by several accessory proteins and makes the cell to function accordingly. Example, phagocytic formation in amoeba or amoeba movement where the actin thread milling is continuously takes place, and that's how the amoeba crawls forward. The other protein is a myosin motor which slides or walks on actin